Hello students, we have already discussed about m13 genome and its life cycle. Now we are going to discuss about how we can develop a cloning vector from this m13 genome. As we all know that for a cloning vector, it may have some general properties because it may have an origin of replication, a multiple cloning site, or a polylinker where we can introduce a foreign DNA, a selectable marker. The selectable marker that helps us to trace the presence of vector or the movement of the vector in various progenies. So, for a cloning vector, it requires an origin of replication, a multiple cloning site, a selectable marker, and so on. So, if you wanted to develop this m13 genome into a cloning vector, we need to introduce these DNA elements into it because a wild type m13 genome doesn't have a multiple cloning site and selectable markers. But of course, it having an origin of replication for replication in prokaryotes or in bacteria. So we need to introduce multiple cloning site and a selectable marker into m13 genome to make it into a cloning vector. So here I'm going to discuss these two things. That is, one is selectable marker and another is multiple cloning site. Selectable marker, while we consider with that of the prokaryotic organism, is laxed. This is a good one and also a commonly used selectable marker because marker we can complete the selection process in one step. That is the advantage of it. That's why in normal cases we are introducing or we are using or we are depending this laxed selectable markers for introducing this laxy properties into a vector. We need to introduce three DNA elements which are associated with the laxed gene. They are an operator proximal region of laxed gene, promoter for that laxed region, and a repressor or regulator region for controlling this promoter region of the laxed region. So we all know about this lac is lac I, lac promoter, etc. because we have studied it in lac operon. This is the structure of a lac operon. This lac operon having a promoter adjacent to it, it having operator. Then downstream to operator, there is a sequence of structural genes. They are laxed, lac Y, lac A, lac I gene that actually located outside of the lac operon. This is actually not a part of this lac operon, but whatsoever this lac I that produces a regulator that requires to control the transcription of these structural genes or that requires to regulate the transcription from the lac promoter. So this lac-I produces a regulator protein. That regulated protein interact with the operator region of the lac operon. This operator region sequences that overlaps with that of the promoter. So if the operator is occupied with the regulator or repressor protein, then promoter, some of the sequence of the promoter may also get masked. Hence, this RNA polymerase enzyme can't recognize this promoter region. So transcription of this laxed lac Y lac A cannot begins. If the regulator proteins or repressor protein is not occupied at the operator region, then promoter may be exposed, then that promoter may be recognized by RNA polymerase, and transcription begins from this promoter. So a polycystonic mRNA of LAC-Z, LAC-Y, and LAC-A get transcribed for imparting the vector with LAC-Z properties. I'm talking about this LAC-Z. This LAC-Z, it produces the beta galactosidase enzyme. That beta galactosidase enzyme normally that cleaves lactose into monosaccharides. In lab conditions, it cleaves an analog of lactose, that is X-gal, into a blue colored indigo dye. So those bacteria having this laxid produces beta galactosidase, and that cleaves X-gal that present in the medium into a blue colored dye. So those organisms can be, those bacteria cells can be seen as blue colored cells or blue colored colonies. Those bacterial cells doesn't have laxid, or having a mutated laxid, they won't produces beta galactosidase enzyme, so it cannot cleave X-gal into blue colored dye, so they won't develop any blue coloration. Hence those colonies can be seen as white colored. Okay. So coming back to this slide, I have already told that if you wanted to impart a vector this laxed properties, you need to introduce these three segments. These three segments means an operator proximal region of the laxed, and another fragment to be introduced is the promoter region of the laxed along with the operator, and next and the third fragment needed to be introduced into the M13 phase is the LAC-I. So LAC-I, promoter along with the operator, then laxed. These three elements are required to introduce into the M13 phages to impart these selection capabilities. Because this laxed or beta galactosidase is transcribed from this promoter, and this promoter is controlled or regulated by a regulator protein that is produced from LAC-I. Okay, so these three elements should be introduced into M13 phages. Next is the multiple cloning site or polylinker. What is a polylinker? A polylinker is a short DNA sequence or short DNA fragment having unique restriction sites. See, this is a short DNA fragment having unique restriction sites. Here in this short DNA, the restriction sites are ECOR1, SAC1, KPN1, SMA1, AMH1, XBA1, SALV1, PST1, SPH1, HIN3. All these restriction sites are present within a short region of the DNA, and these short regions of the DNA, altogether these restriction sites are known as multiple cloning sites. So you have to introduce a multiple cloning site also into the M13 genome. So this is a vector constructed from M13 phage, that is M13 MP18. Here in this case, you have introduced a LAC-I region, then promoter for laxed, this red arrow, and this purple arrow starting from here to here indicates the laxed region that you have introduced into M13. And in addition to these three elements, I have told you that you had to add multiple cloning site also into M13 patch. This is the multiple cloning site or polylinker. this black region. See, starting from here to here, this is a polylingular or multiple cloning site. For introducing this multiple cloning site over here, you need to cut open the lax region of the DNA with the restriction enzymes. Once it opened, then you have to introduce this multiple cloning site into that open region. Okay, the first thing is that this is the lax region. Into that reading frame, you have to cut open that reading frame, or you have to cut open the laxed gene, and into that gene, into that restriction open region, you have to add, or you have to negate a polylingular over here. Okay, here lies a complexity. You know why? Because if this is the polylingular, it can introduce into this particular region in two opposite orientations. For example, uh, this is the polylingular that we have, or that are present in the MP18 vector. That means it starts from echo R1 and ends with the hin 3. Okay, that means here in this case M13 MP18, it starts with the echo R1 and ends with the hin 3. Okay, start with the echo R1 and ends with the hin 3. This is one way. In another way, also it can be introduced into this region, that is in an opposite orientation. That means there it starts from hin 3 and end with the echo R1. Exactly opposite of the orientation that I have told just a while ago. Okay, I think you have got that. That means in one way, it inserted in such a way that echo one starting from here and ends with the hin 3, and in another orientation, echo one ends with the here and hin 3 start from here. Okay, so they're created. So there we have created two types of vectors, because it having a small variations at polylingular region. In one vector, it's in a particular orientation. In another vector, it's in an opposite orientation. So in one vector is known as M13, MP18, and another vector having an opposite orientation to the polylingular, that is M13, MP19. So we have constructed two vectors over here, that is M13, MP18 and 13 MP19. Both of them having only one difference at that is at the polylingular region. These polylingular regions are oriented in opposite ways in both of these. So here it indicates these two polylinguals. This is of MP18 starting from echo R1, SAC1, KPN, SMA1, BAMH1, SBA, SAL, PST, SPH, HIN3. And in MP19, HIN3, SPH, PST1, SAL1, XBA, BAMH1, SMA1, KPN, SAC1, Coron. Exactly the reverse of this. Okay. Coron comes over here and here also. SAC1 comes over here and here also. SMA1 also, sorry, KPN comes next in both of these cases. Okay, and so on. Next, I'm going to talk how we can clone a foreign DNA into this M13 vector. For that, you need to isolate double-stranded M13 vector or M13 phage. I have already told you that this M13 phage or M13 vector that exists in two forms within the bacterial cell. Here, double stranded form and single stranded form. Double stranded form is known as RF or replicative forms. These replicative forms are much more important in the cases of cloning experiments, because for cloning experiments we need to cut open the vector with the restriction enzymes. But the restriction enzymes may recognize as only double stranded DNA. It cannot bind and digest single stranded DNA. That's why you need to isolate M13 in RF form, and then you have to cut or digest it with any one of these restriction enzyme. So you will cut open the multiple cloning site. Then you have to introduce the foreign DNA into this multiple cloning site. This can be of two orientations. One in MP18, it may be in one orientation, and in MP19, this orientation of the inserted DNA may be in X side. And in MP19, the orientation of the inserted DNA may be exactly reverse that of the MP18. Okay. So next, why do you need this M13 vector? These M13 vectors are much more important for getting single stranded DNA for the purposes of sequencing and mutagenesis. That is, in the case of sequencing, especially Sanger's dideoxy method, we need a single stranded DNA. And that DNA will be amplified in the presence of DNTP and DDNTP. Then you will get chain terminations at so many places, and hence you can get to know the sequence of the DNA of interest or gene of interest. For that, what you have to do is that, suppose if you want to get to know the sequence of this gene, actually you need this gene as a single stranded DNA for doing or for sequencing purposes. For that, what you have to do is that you have to excise that gene from a double stranded DNA or from the double stranded genome, and this is double stranded gene, and that you have to insert into M13 vector. That is the double stranded form of the M13 vector. Then you will get a vector inserted with your foreign DNA, and this vector need to be introduced into a bacterial cell by means of transformation. Then during its life cycle, that bacterial cell may harbor both single stranded and double stranded forms of this vector. If you wanted single stranded form of this gene, then isolate single stranded vector, and that vector can be used for sequencing purposes using primers against vector adjacent to the inserted region of the DNA. So in the case of mutagenesis, side-directed mutagenesis also we requires single-stranded DNA. Actually, this side-directed mutagenesis is a long lesson that we will study later. So we have developed two vectors, that is M13 MP18 and M13 MP19. Next, we are going to develop some more vectors from M13. They are M13 MP1, M13 MP2, and M13 MP7, and so on. So for that, I'm going back to the M13 genome. Actually, this M13 genome is quite complex in nature because its life cycle is quite complicated. because you know that M13 is a bacteriophage. Especially this is a temperate bacteriophage. So this can switch in between two life cycles. They are lysogenic cycle and the lytic cycles. For each of these cycles to be carried out, they are having their own genes responsible for each of these cycles. I think you may have heard about immediate early genes, late genes, immediate early genes, etc. So while we compared with that of a prokaryotic or plasmid vectors or a bacterial genome, this phage genome is quite complicated or complex because they're having a complicated life cycle. That is, some of the phages having a lysogenic cycle, and some of them having lytic cycle, and some of them can opt or shift in between these two. They are known as temperate phages. This M13 phages is an example of a temperate phage. So its genome is of obviously quite complicated. See, this is the genome of M13. We have already had a detailed look at on this before. This is gene 4, this is gene 1, and so on. These Roman letters, all of them are indicated different genes. Those are present in the M13 genome. Actually, you won't have much choices to alter these regions. You know why? Because these are coded regions, and all of these genes are important for the phage life cycle. Suppose if you insert or alter the sequence over here, then definitely this gene form may get damaged. Then it definitely have deleterious effect in phage life cycle. So you cannot modify these regions. Okay. But luckily we have found that a small region over here in between gene 4 and gene 2, an intergenic region. Intergenic means in between two genes. So an intergenic region in between gene 2 and gene 4. This is the intergenic region. See, this is the intergenic region, and this intergenic region having a length of around 507 nucleotide. Actually it doesn't code anything. That's why we will get choices to modify these regions. But of course you have to think about one thing, or you have to keep in mind that this intergenic sequences, it calls for origin of replication. You cannot touch over here, because this is something, a sequence of very crucial in nature, because you know that this origin of replication is important. You know that this origin of replication is important for this vector replication, because these regions can be recognized by DNA polymerases, and replication may start from here. If you alter these sequences, then definitely that impairs the replication of this vector. So that won't be a good protocol. So that's why I'm telling you that if you wanted to modify this M13 genome, you have to modify this region, this intergenic sequences devoid of this origin of replication. Okay, so these all these regions that may be available for modification, except that this origin of replication. Okay, so next we are going to prepare a vector that is M13 MP1. Okay, see this picture. This is gene 4, this is gene 2, and this is the intergenic region. This is exactly similar to that I have explained just a while ago. Okay, this region for origin of replication, IS, and gene 2. This is exactly similar to that of the just mentioned thing. And what you have to do is that you have to introduce a selectable marker that is laxed at over here in this intergenic sequence. Okay, so you have to cut open here with some restriction digestion protocol, and then you have to introduce a laxed gene over here. So this is the modified m13 genome having laxed selectable marker. So now you have developed or constructed a vector, and this vector is known as m13mp1. That means a phage genome having one laxed region. Okay, this is m13mp1, but definitely it's having so many drawbacks because it doesn't have any restriction sites. So you have to construct another vectors from this mp1 having these additional features. So for that, see, this is the m13 mp1 just we had prepared a while ago, and you have to make two changes over here for developing m13 mp2. That means you had made two changes over here for developing m13 mp2. One change is that this laxed region having a sequence somewhat similar to this. This is the beginning of the laxed region. Okay, can you see this region? That is G G A T T C. This sequence is similar, somewhat similar to that of a codon restriction site, but it having one nucleotide varied from that of the ECOR1 restriction site, that is at this place. This ECOR1 restriction site having a sequence of G A instead of G, so G A T T C, that is ECOR1 site, but here in the laxed region we having G G A T T C sequence. So here we having a sequence similar to that of restriction site. So for developing a restriction site over here, what you have to do is that you have to create a point mutation at this place. So you have to uh, mutate this G into A. Then this particular sequence may be converted into a codon sequence. So by means of site-directed mutagenesis process, you can introduce such mutations over here. So here you can create an ECOR1 site over here. Okay. So by means of a point mutation, you had created an ECOR1 site over here. And the second modification for constructing M13MP2 is that you have to introduce a and the second mutation that you had introduced into the MP1 for constructing MP2 is at codon 6th of the laxed. Normally, the codon 6th in this laxed gene that calls for aspartic acid. Here for constructing MP2, you had altered that codon into a codon for asparagine. So here in MP1, the sixth codon that may be changed into a codon of asparagine instead of aspartic acid. These are the two alterations that we had made in MP1 for constructing MP2. So first one, you had created a restriction site over here. Second, you had changed sixth codon into aspartic acid codon. So there develops a vector. That vector is known as M13 MP2. Now you are going to construct another vector, that is MP7, because this M13 MP2 having some more drawbacks over here. You know why? Because it's having only one restriction site over here. So this may not be a good choice on all cloning activities, because if you had excised a foreign DNA by means of a different restriction enzyme, for example HIN3, so you cannot introduce that DNA fragment into this particular site, because that DNA fragment is not at all compatible with the occurrence site. So what you have to do is that instead of this echo R1, you are going to introduce a multiple cloning site or polylinker over here. Okay. So for that, for constructing MP7 from MP2, what you have to do is that you have to cut this DNA, or you have to cut this laxed region with a coron enzyme. So this coron site may get opened, and into that region you have to ligate a polylinker or a multiple cloning site. So this MP2, now it may have a polylinker or a multiple cloning site at over here. Now it may not be called as M13 MP2. Now this is a different vector, that is M13 MP7. Uh, See the sequence of the polylinker we have introduced into MP2 for constructing MP7. This is the sequence: echo R1, BAMH1, SAL1, then after that it is PST1. Okay. Coming to the disadvantages of M13 vector. This M13 vector having some disadvantages. The most important disadvantages of M13 vector is it can occupy only a limited foreign DNA. That is around 1500 base pair of DNA. Only that much of DNA can be introduced into M13 vectors. In recent generations of M13 vectors, we can introduce up to a maximum of 3000 base pair or 3 kb of foreign DNA. So this is a much smaller DNA that can be that we are introducing here. So obviously this is a big drawback of this M13 vector. Okay.